Good morning again. This is Science Activate Week 4. We're going to look at the last batch of questions. Hopefully you guys have done pretty well on the last couple of weeks. So here we go. Number 1. This question looks at experiments. All right, we have two different experiments, and one of these experiments is not experiments. These are two different viewpoints, and these two different viewpoints are about DNA and about protein this week. So these are two scientists who are talking about DNA versus protein. So the DNA hypothesis states that genes are made only of DNA, and that DNA is found exclusively in the cell's nucleus, whereas proteins are found throughout the nucleus and the cytoplasm. Additionally, the amount of protein in the cell varies. So protein in the cell varies from cell type to cell type, even within the same animal. Though DNA is less abundant than proteins, the amount is consistent from cell type to cell type. Amount is consistent. So they're saying that protein isn't consistent, but DNA is, except for gametes. Gametes are the reproductive cells. Gametes have half the amount of DNA as other cells in the body. Gametes also have half the typical number of chromosomes. Thus, the amount of DNA in a cell is correlated with the number of chromosomes in the cell. No such correlation is found in proteins. So they're saying that DNA is in charge of the genes of our bodies and of animals and of humans and of everything. And so the question says, which of the following statements is most consistent with the DNA hypothesis? The amount of DNA will generally increase from cell type to cell type as the number of what? Is it the amino acids? Well, if you know anything about proteins, you know that amino acids are in proteins, so we can cuss that out. G also says amino acids. So if you didn't know that, though, you can look at H and J, and we say chromosomes. There's chromosomes here. All right, so it says the amount of DNA in a cell is correlated with the number of chromosomes in the cell. Gametes have half the DNA and half the chromosomes. All right, so we're looking at H versus J, and it says chromosomes in the nucleus increases from cell type to cell type, or chromosomes in the cytoplasm increase from cell type to cell type. So where is that DNA? Where are those chromosomes? Well, we said up here, DNA is found exclusively in the cell's nucleus, and proteins are found in the nucleus and the cytoplasm. So if we're just looking at nucleus, H is our correct answer. Number 2 looks at the protein hypothesis. All right, so our protein hypothesis says genes are made only of proteins. That's their statement. They say that's what it is. Proteins make up 50% or more of a cell's dry weight. Cells contain 20 different amino acids that can be arranged in virtually infinite number of ways to make different proteins. So different proteins from 20 amino acids. The number and arrangement of different amino acids, they're saying that forms the codes that contain hereditary information. Now this is just a hypothesis, and I want to stress that we know that this is not true, but these are two different differing viewpoints in this experiment. In contrast, only 4 different nucleotides make up the DNA found in the cells. They are believed to form chains only in certain ratios. As a result, the number of different combinations that DNA can carry is much smaller than the number of proteins can carry. So they're saying that there's way more option for different types of coatings with proteins, and they're saying that the DNA has a much more limited, because there's only 4 nucleotides in DNA. So let's look at the protein hypothesis question. According to the protein hypothesis, which of the following observations provides the strongest evidence that genes are not composed of DNA? All right, so the strongest evidence that they talked about over here is that the DNA is composed only of 4 types of nucleotides. that was a pretty strong with 20 versus 4. That might be the right answer. DNA is composed of smaller subunits than our proteins. That didn't really get stated, and it doesn't really work, so we can get rid of that one. DNA is abundant in both the nucleus and the cytoplasm. Well, we know that that's not true, so that doesn't really support our argument. And this one says the concentration of DNA is generally consistent from cell to cell. This didn't say anything in the protein hypothesis about that, so it must be A, the DNA is composed of only 4 types of nucleotides. We know now that the nucleotides actually form those amino acids. They help to code for those amino acids. So we know that there are lots of other chains like that. But that is question 2. DNA is composed of only 4 types of nucleotides. Question number 3 this week. We have Experiment 1. All right, so we're now away from those 2 scientists' differing viewpoints, so we're back to an experiment. And so in Experiment 1, we have sucrose broth added to 5 large test tubes. Phenol red, which is a pH indicator, is yellow if pH is greater than 7, red if pH is yellow if pH is less than 7, and red if pH is greater than or equal to 7, was added to each large test tube. A Durham tube, which is a small test tube, was placed inverted. There's an inverted Durham tube right there in each large test tube to collect carbon dioxide. So if we see a bubble, we'll get carbon dioxide. So that's where these come in over here. If there's CO2 and it's a plus sign, it means there's a bubble. If there's acid, you need to know that pH less than 7 is an acid. So yellow means acid. Okay. The large test tubes were capped, heated until the solutions were sterile, then cooled. One of 4 bacterial species, A through D, was added to each of the 4 test tubes. The procedure was repeated using lactose broth instead of sucrose broth. The 10 large test tubes, all containing solutions at a pH of 7, were then incubated at 37. So 7 is neutral, and they were incubated at 37 for 48 hours. The large test tubes and Durham tubes were examined. If acid was produced, the solution was yellow. So yellow means acid. If no acid was produced, the solution remained red. No acid. All right. If CO2 was produced, a gas bubble. So a gas bubble means CO2. Was observed at the top of the Durham tube. All right, so in Experiment 1, which of the bacterial species fermented lactose? So which one had a reaction with lactose? If we look over here at our lactose broth, we have 2 species. We have an acid fermentation on both of these, and we have a carbon dioxide fermentation on this one. So we have species B and species D that both did something here. They both had a fermentation. So when we fermented lactose, we end up having species B and species D only. There's question number 3. All right. Question number 4. We got the same information here. But suppose a scientist isolates a bacterial species that is one of the 4 using Experiment 1. She adds the species to sucrose broth and observes that neither acid nor CO2 is produced. So if she adds it to sucrose and neither acid nor CO2 is produced, it can't be C or D because they both have those. She then adds the species to lactose broth and observes that both acid and CO2 are produced. So she adds one of these to lactose broth and she observes both lactic acid and CO2. So this must be B. Based on the results of the experiment, the species is most likely species B. All right, let's look at the last question here. Question number 4. It looks like we got some Durham tubes to actually look at. So we've got some yellow broth and some red broth. Let's look through here. Here's our table again. So what's our question say? Which of the following best illustrates the results of experiment 1 for species D in the sucrose broth. So sucrose broth, experiment 1, species D. So species D is a positive acid and a negative CO2. So we need, if remember, yellow is acid and CO2 is a bubble. So no bubble and yellow. Which one has no bubble and is yellow? Well, this one has no bubble and is yellow. So G is our correct answer for question number 5. Thank you guys. I hope you have a great time taking the ACT. You got lots of good training here. If you have any questions or you need some help on your preparation, you can come see Mr. Stewart during Smart Lunch. Have a great time.